This presentation is focused on a new product which is deriving from the combination of a bergamot extract we discussed in the last few years, and a wild type wild cardoon extract, which is an original composition since it's a product by process. It's not just a combination of two powders, as many of the extract you can find around, but is exactly deriving from the interaction of the liquid phase of the two products originating the one you can find in the final raw material. And the target we chosen for this product is the so-called fat switch. So there is a huge interest in detecting not only which are the characteristics of the endemic evolution of obesity in Western, but only in Western population. And you can see that 2.5 billion of people are today suffering from metabolic syndrome and obesity. And in this area, the localization of the fat in the liver seems to be a crucial point, since this affects the development of cardiometabolic risk. And you can see that this stage is called the non-alcoholic fat liver disease, but we know that the development of fibrosis and inflammation is a crucial key point in the development of the disease. The disease is mostly related to the, it is known, lifestyle changes, which have been found in the last 50 years. And you can see that our performances in terms of controlling the increase of fat in Western population is mostly related to the lifestyle changes, including overstressing condition, lack of sleep, and inflammatory components you can find in many of the foods which are consumed in restaurants or in the fast food. This is not only obesity, but as you know, we have a huge correlation of obesity with cardiometabolic disease, and you can see that the 40%, the 44% is associated to type 2 diabetes, the 33% to ischemic heart disease, but there is increasing evidence that increasing fat is associated to the increasing occurrence of cancers. So, which is the key pathophysiological event which may be eventually approached and controlled by drugs or nutraceuticals? We believe that excessive formation of free radical species represents a key mechanism which is underlying all the changes occurring in fat tissue. We will evaluate, especially the development of inflammation and the alterations occurring in adipose tissue are the one which we can define the fat switch. And we will see that this is not evident from the morphological point of view, but serves functional changes related to the type of fat which is activated. And these are the friends, insulin resistance, cardiovascular disease, and NASH, which are accompanying the development of oxidative stress. The other component is exogenous factors, such as pollutants you can find in environment, but also in foods, which may enhance the inflammation of fat and the development of whitening process which are occurring in the visceral fat. And you can see there that in this area a crucial role is played by microbiota, which is enhancing the development of liver dysfunction and is the key event correlating gut dysbiosis with hepatic alterations and fat alterations and brain disorders. So you can see it's a huge area in which we are going to identify which are the key biomarkers to be followed and which play a crucial role in fat inflammation. You can see that we are switching from an anti-inflammatory normal profile into a pro-inflammatory profile in which cytokines and adipokines play a role in mutating the phenotypic alterations you can find in the fat tissue. And in this respect, we believe that the switch from the white adipocyte to the brown adipocyte or vice versa is a key mechanism, which is the one we believe to may access through the use of several nutraceuticals, including Bergacyn. And especially you can see that white adipocytes is the one which is characterized by pro-inflammatory and promoting cardiovascular dysfunction compared to the brown adipocyte, which are the one we have in the interscapular area, in which the increase leads to protection against cardiometabolic disorders. And this is related to the increased lipogenesis, which is related to the increase of white adipose tissue, increased inflammation, and several stressors which act mostly at the sarcoplasmic reticulum level and at the mitochondrial level in the cells. So we believe that antioxidants may represent a potential supplementation able to produce benefits if associated to anti-inflammatory properties in adipose tissue inflammation. Which one? So many of the products are claimed to may interact with increase of decrease in fat mass, but we know very well that we have not only to reduce the weight, but also to increase the brown adipose tissue in order to have successful response. In the last few years, we studied the bergamot extract, which has been shown to produce beneficial effect in metabolic syndrome. This has published in many papers, including scientific reports of Nature, in which we demonstrated that accumulation of fat in the liver and fibrosis are antagonized by bergamot polyphenolic fraction deriving from this citrus species. And the major characteristics of BPF are the phytochemical uniqueness, since we have selective polyphenols which are at very high concentration, better absorption and tissue distribution, and a well-characterized mechanism of action. We published more than 60 papers in the last 15 years, and we have more than clinical studies which proven the efficacy and the safety of the product. This is the analytical profile which was published by our group in collaboration with one of the most referenced groups worldwide, in which you can see that we have a series of polyphenols which in total get the level of the 75–80% of the extract. And this is the area in which selectively the bergamot grows, but we have seen recently that in the same area we have another player which may be of benefit in trying to managing the fat switch, which is the wild cardoon artichoke extract, which is characterized by very high level of several active ingredients which have been found able to interact with the fat tissue, but also to control insulin resistance. And recently we have seen that there is the ability of this extract to burning the fat tissue and to move to switch on the promotion and the growth of the brown adipose tissue. And this is the major classes of active ingredients you can find in wild cardoon, in Calabrian wild cardoon, and you can see that we have also flavonoids, very important, such as luteolin, but also sesquiterpenes, such as cynaropicrin, which are the one responsible for liver protection of this product, and the product derived from the caffeic acid, which are in a very high concentration and which are the one which stimulate GLP-1 receptors, leading to the reduction of body weight. So you can see is a complex of active ingredients which are mixed together in just one extract, and which have been studied through clinical studies, one having metabolic syndrome and obese subjects as a target, 80 subjects on treatment with current therapies, in which we evaluated both body weight reduction and the liver protection in terms of development of fibrosis. And this has been published in Frontiers of Endocrinology, in which you can see that the reduction in body weight was associated to a substantial reduction of liver fat as measured by the means not only of ultrasonography, but also detecting the fibrosis score. We demonstrated that the fibrosis score is reduced when approaching with Bergacyn, including fibers from bergamot, which enhance the absorption of active ingredients. The second study was carried out in patients with diabetes mellitus and non-alcoholic fatty liver disease, at the stage in which the level of fat in the liver is consistent, and you can see that we have a huge reduction through the Bergacyn of liver fat, but especially we resolved the major marker of the compromised cardiovascular response, which is the endothelial dependent relaxation and the release of nitric oxide. You know that nitric oxide is important for maintaining the vasodilatation in blood vessels, and the index is the RHI reactivity index is improved in subjects, in diabetic subjects with fatty liver, in which Bergacyn was used. And all the other components related to oxidative stress and inflammation were modified and improved in subjects taking Bergacyn. This is the overall biomarkers which are improved by bergamot, chinara, and bergamot fibers. Then we very recently found that the crucial point is the switch from white to brown adipose tissue. This has been carried out in a special model or animal model of rats developing obesity and insulin resistance. Only 2 groups worldwide are using this model, as this model is characterized by mimicking completely the development of fibrosis, liver fibrosis in humans, and is the only model admitted by Food and Drug Administration for novel foods and novel drugs eventually. And in this model, we evaluated the effect on lean mass and fat mass. You can see that we have a significant response, that we have a reduction of fat mass, and this was associated to an increase in brown tissue you can find through MRI in the interscapular region, as shown by the arrows in the figures, and you can see that brown adipose tissue was increased compared to the reduction of white adipose tissue. This was accompanied by significant reduction of biomarkers of oxidative stress and inflammation, and this was mostly related to the modulation of transcription factors, which are a key element in the chain of activating browning of adipose tissue. PPAR gamma is the principal one when NFKD was reduced, as indicating that we have an anti-inflammatory response. So, it's okay. In conclusion, we demonstrated that Bergacyn, which is a combination of bergamot extract, especially the BPF extract, which is the special composition of bergamot polyphenols, with wild cardoon extract, is able to produce an anti-inflammatory and antioxidative stress response in liver tissue of obese subjects. And the additional information is that this extract is able to interact with the qualitative composition of adipose tissue, increasing the brown tissue, which is protective and cardioprotective, and reducing the white visceral adipose tissue, which is the responsible for inflammation and cardiovascular dysfunction. Thank you very much for your attention. Observe. Any question is welcome. I have not the microphone. People told me that I have to manage.
Thank you. Did you test any of the, because you said that there's improved glucose homeostasis. Was that tested in any of the clinicals? Did you use oral glucose tolerance test?
Sorry?
For the improved glucose homeostasis in the clinical trials in diabetics and obese patients, did you test them with oral glucose tolerance test?
Well, we had both an improvement in the insulin response in terms of fasting glucose. I didn't display all the data. And also in terms of glycosylated hemoglobin, which is totally reduced. Also in patients, we had an improvement. And I explained it during the presentation that this seems to be related to activation of GLP-1 receptor. You know, GLP-1 receptor are the one responsible for body weight response in normal subjects, but also especially in diabetics. And you have also an increase of the browning. This is lacking for many of the GLP-1 receptor agonists you can find in the market, liraglutide and so on.